Welcome to this Advanced A&P lab at Western Technical College for genetics and heredity. This one was not shot in front of a live classroom. We did have, unfortunately, a snow day, so this one I'm going to be doing just as a voiceover here today. And so we'll start here by looking at Objective 1 and just kind of explaining the following. What is a chromosome, which is basically a set of chromosomes, which are 23 very unique chromosomes, and we have a set of them. So under normal conditions in a body cell we have 46 chromosomes, but you inherit 23 of those chromosomes from mom, 23 of those chromosomes from dad. And so prior to that, your mother gave you essentially one set, which if it is 23 chromosomes, that would be a haploid cell. So the mother's egg would be an example of a haploid cell. It had 23 of the chromosomes, and then the father's sperm had the other 23 of the chromosomes, so that would be a haploid cell as well, being that it has only half of the chromosomes. Where in a normal body cell, we would call that that has 46 chromosomes, that's considered to be a diploid cell. And so our body cells are also known as somatic cells as well. And so when we look at the chromosomes that we get, we typically look at them as one from dad, one from mom, that are said to be homologous, meaning that they are supposedly supposed to be the same chromosomes, if you will, but they may express different traits, if you will. And we'll look at that as we go along. And so you have 23 chromosomes from dad, 23 chromosomes from mom. The first 22 chromosomes, if you will, are going to code for what we call body traits, and this is the autosome chromosomes 1 through 22. And so any type of autosomal linked trait is going to be a body trait determined by the genes on an autosome. And so if you look over at the picture that I have in this slide here, you're going to see all of the 22 autosome chromosomes there until you get to the very last two, which is way over at the end, which is your X and your Y, or in a female might be X and an X. And so these last, the 23rd chromosome, if you will, or set, if you will, is going to be your sex chromosome, which is our next one here. And so some body traits also are gender traits, such as this X and X, which would be for the female. If you saw an X and a Y, that would be for the male. And as you can see just with that picture, the X and the Y with the male, being the male has a different chromosome there as the Y chromosome, you can see that they are not homologous. They are different. And so you can see that in a male that all of the other chromosomes are going to be homologous except for the X and the Y, the sex chromosomes. Now in a female they have an X and an X, which would be homologous, but not the case for the male there. And so sex-linked traits are body traits that are determined by that 23rd chromosome, the X or the Y chromosome, depending if you're looking at the male or the female. So just kind of recapping probably some things that you're familiar with from past classes. Chromatin is a fancy word for DNA, but we find chromatin in a non-dividing cell, and typically chromatin or the DNA in this format is very stringy and expanded out or stretched out, if you will. And then when the cell is going through mitosis, it supercoils or condenses up into what we call a chromosome. And a chromosome is also a fancy word for DNA, but found in a dividing cell. So it's just a wordsmith thing there that you see there. DNA is DNA, but when it's in a non-dividing cell, we call it chromatin, and when it's in a dividing cell, we call it chromosomes. And so if we just look at chromosomes, there are several different parts of it that you need to be familiar with, and hopefully this is just a recap from General A&P. But if we go over to this picture here on the next slide, you're going to see the cell there and the nucleus in the middle, and we've expanded one of those chromosomes. And now it's very supercoiled there, and it's in an X-shaped format because it's going through division, and so one of those chromosomes there is identical to the other one on the other side. So essentially, if you split this chromosome into two, they really are identical DNA matches, if you will. But when the cell's dividing, it tends to supercoil it up and make it look like this X-shaped format. Now, each one of these halves, if you will, are considered to be a sister chromatid or a chromatid. So this would be a sister chromatid, this is a sister chromatid, and they call them sisters because essentially they're identical, if you will. Now, in the middle of the chromosome, which you don't really see in this picture here, but you see it over here, there is a protein called a centromere. And the centromere is what's holding these two chromosomes together before they actually split apart. And when we look at this chromosome, typically we see a short arm, which is the P, and then we have the long arm, which is the Q. And so if we go over here, you can see that these are some things that you need to know. That short arm is the P arm, and the long arm is the Q arm as well. And so these are daughter chromosomes, if you will, because what they're going to do is split apart, and that takes place, if you remember back to mitosis, during the anaphase phase of mitosis, where this chromatid here is going to split apart from this one, and they're going to go to their respective sides of the cell so that it can create two new daughter cells. And so, you know, if you look at that P arm and that Q arm, and you can see those color differences there, those are all of your genes, and some of them are important and some of them aren't important. It just depends on which ones they are. And I don't want to get into too much of that right now, but we'll kind of get into that as we go along. But all those discolorations that you see there, essentially those are what you would consider your genes. Now remember, genes are responsible for coding for specific proteins in your body, and that's what these are going to be doing. So there we have it. The gene, as we just talked about, a gene is going to, it's the blueprint of your body. It's going to specifically, though, code for specific proteins in your body, and these genes are on different what we call gene locus, which is what I was referring to earlier, which is the very specific location of the gene. So as an example that we have here in parentheses, it might be the Q arm, and it would be chromosome number 7, and so that's kind of where we would look on that particular chromosome to find that gene that might be a specific gene for a specific thing that our bodies create in our body. Now, alleles are partners of gene pair, so they could be the same or they could be different. You get one from mom and one from dad. So remember, dad is contributing 23 chromosomes, your mother is contributing 23 chromosomes. And if we go back to this picture here, right there, these are the 23 chromosomes. So they should be, you know, identical, if you will, as far as the chromosomes, as far as the length of them, but the genes may be different, and that's kind of what we're getting into right here, is these alleles, these are partners of gene pair. So your mother's colored eyes compared to your father's colored eyes will be on the same gene pair, but of course your father's or your mother's may be dominant over the other. We'll get into that here. The dominant allele essentially masks or suppresses the other genes. So if your dad's eyes are brown and your mom's are blue, and your dad's are the dominant allele or pair or gene pair, your dad is going to suppress your mother's recessive gene, and your recessive allele is the allele that is being masked or suppressed. So you always have, not always, I shouldn't say that, but a lot of times you have one that's going to be more dominant than the other one, and that dominant one is going to override the recessive one. Now that is not always the case, as we will see here moving forward, but just real basic information there. So a genotype are the gene pairs that are inherited by an individual. So that's kind of what we were talking about earlier, these alleles, these gene pairs. And so sometimes the gene pairs or alleles may be homozygous genotype, which means that the alleles are the same, so you can see in parentheses. And by the way, anytime you see a capital letter, that means dominant. So this homozygous genotype here being the same It has two capital C's. Whereas if they're different, if you have two alleles that are different, like you have a dominant and a recessive, as you can see here with a capital C and a lowercase c, that is heterozygous genotype. And that's the genotype and how the genes are paired and how they're inherited by an individual, whether or not they're homozygous or they're heterozygous. But then the other part of that is how is it being expressed? And we call that the phenotype. So do you get brown eyes? Do you get blue eyes? That's how it's going to be expressed, right? So you might have this genotype, but then it's like, okay, well, what exactly is going to happen to it now that we have them, and how is that going to be expressed? And that's what we call the phenotype. Do you have brown hair? Do you have blonde hair? Light skin, dark skin? Those kind of things are what we consider to be the phenotype. How is it expressed? So we do have differentiate between the following here. Monogenetic inheritance, that's where a body trait by one gene and a single pair of alleles. So, just one particular gene is only going to express, or one gene which has two alleles, right, one from mom, one from dad, is going to express that body trait, and that's what we call monogenetic inheritance. So remember, mono means one, so that's single pair. But then there are also different types of inheritance called polygenetic inheritance. So poly means excessive, so we get body traits by several different gene pairs and several alleles that create this what we consider to be polygenetic inheritance, which is a very highly variable phenotype. I believe skin falls under that one. We have so many different shades of skin amongst all of us that that is coming from a variety of different body traits within our genes to create that. It's not necessarily a single gene on a single pair of alleles. It's several different alleles that are creating that skin tone, if you will, and that's polygenetic inheritance. Okay, so now we look at just some simple dominant recessive inheritance here. So if the alleles are both dominant alleles, then the dominant trait would override that, and because they're both dominant, obviously. Now if both of the alleles are recessive, then it would be a recessive trait. But if we have a dominant and a recessive, then the dominant is going to override the recessive, and therefore it would be a dominant trait. That's not always going to be the case as we get into this a little bit more. There's, of course, a lot of stuff to this, but if we were just looking at what we consider to be very simple dominant recessive inheritance, it would be very straightforward. But, of course, we're going to have some gray area, if you will, and you'll see that in just a moment. So a genetic carrier, this is where you get that if you have a heterozygous genotype, which means that you have a dominant and a recessive, right? And where this recessive allele is being masked by that dominant allele. So even though you have this recessive allele, this means that you carry the recessive trait, which means you can pass it along to your offspring. Okay, so if you're a heterozygous genotype, you have both a dominant and recessive. Even though the dominant is masking the recessive, and since you have the recessive, that means that you're carrying the recessive trait. So just keep that in mind, because you will see that we'll be passing that along to the offspring, and that could come into play later down in the gene pool, if you will. Okay, so here's where we get kind of that gray area that I was telling you about with dominant. It's not as easy as if you have two dominants, then it's a dominant. You have two recessive, it's going to express as a recessive, and if you have a dominant recessive, it's going to express as a dominant trait. And so here we have codominance, okay? This is where two dominant alleles that are different. Interesting. So here, as an example, your blood type. So if you don't already know, there's different blood types, A, B, AB, and O. And so you can see you have blood type A, you have blood type B, and then, we have blood type AB. Why do we have that? It's this codominance, where we get these two dominant alleles that are completely different, but they're going to create this very unique phenotype, how it's expressed again, being a B blood type, okay? So that's an example of this codominance, where you have two that want to be dominant, but they're different, and they just kind of merge together to create this very unique phenotype, in this case, blood type AB. Now, incomplete dominance is where you get heterozygous genotype, you get a dominant and a recessive genotype, and the phenotype, how it's expressed, is actually between both of them. So normally if we had a heterozygous genotype, a dominant recessive, like I talked about earlier, the dominant would be expressed, it would be a dominant trait. But in incomplete dominance here, we get a little something in between there, and the phenotype is a little bit of both. And so what happens here is that the dominant doesn't completely mask the recessive. Okay, so an example would be sickle cell trait. You have normal blood cells, you have sickle cells, and then you have sickle cell trait, and that trait is in between there, where you know, if it was normal, you'd have normal blood cells. If you had sickle cells, you'd have sickle cells where the dominant would be overriding the recessive, and you'd have sickle cells. And in this case, it's kind of an in-between, where then you get a sickle cell traits, which means that you make some healthy red blood cells and you make some sickle cell blood cells as well. So you have a little bit of both in that case. So this is an example of incomplete dominance. Now, I will let you go ahead and work on the worksheets that we provided you with the Punnett square. And if you have any questions, please let me know.